So hello again. In this video, I want to talk about our paper in which we showed that structural MRI and functional MRI differentially predict chronological age on the one hand and memory performance on the other hand in older adults. So just to give you a little background on this, cognitive performance typically declines with increasing age, but nevertheless, some older adults still show cognitive capabilities comparable or even superior to that of young subjects, which is here shown for memory performance. These older adults are called successful ages, and it is yet an open question whether this successful aging is due to preserved brain structure, what one could call the brain maintenance view, or whether it is due to functional network activity reorganization, which could be termed the cognitive reserve hypothesis. Although we cannot decide between these two theories with our study, the different hypotheses do make different predictions about the correlates of memory performance. So whereas according to the brain maintenance view, differences in memory performance should co-occur with differences in structural MRI patterns. If the cognitive reserve hypothesis is correct, then differences in memory performance should be correlated with differences in functional MRI responses. So we took 106 young subjects aged 18 to 35 and 153 older subjects aged 50 plus, and we measured their brain activity while they were watching photographs showing scenes that were either new to them at the time of presentation or which they had seen before a couple of times, here called master images. So in a retrieval session, 70 minutes later, outside the scanner, subjects were then asked to rate all the old images as well as a couple of new images on a 5-point scale ranging from 1, definitely new, I've not seen this before, up to 5, definitely old. I do remember this from the encoding session. This then allowed us to infer on brain activity differences with respect to novelty processing by contrasting novel images with master images, here shown on the left, and to infer on subsequent memory effects by contrasting later remembered with later forgotten items, here shown on the right. And from these fMRI contrasts, one can also extract single value scores that indicate the similarity of a given older adult's brain activity with the brain activity of a reference group of young subjects. And you can learn more about these scores in a precursor to this paper and in an associated video that I also made. So we took our fMRI contrasts indicating activity associated with novelty or memory, as well as the fMRI scores extracted from them, as well as behavioral data, that is response frequencies in the retrieval session. From each subject, we also measured a structural MRI scan from which we extracted voxel-wise gray matter density, as well as a resting state fMRI session from which we extracted voxel-wise amplitude fluctuations. We used all these feature sets to predict a number of variables, namely to which age group a subject belongs, chronological age in years, and memory performance measured as area under the curve. So first, we found out that age group can be classified based on all these variables with a clear hierarchy from behavioral over functional up to structural information, which holds the most information about aging. If we then look at the continuous variables and here focus on the older adults, we see that chronological age is best predicted from structural MRI, whereas memory performance is best predicted from functional MRI and especially single value scores. So in order to follow up on this, we did a subgroup analysis by partitioning the older subjects either by chronological age into young and older subjects, or by memory performance into older subjects with lower versus higher memory. Doing so, we found that there's a double dissociation in the sense that when partitioning older subjects by chronological age, there are no significant effects with respect to functional MRI, but strong differences in gray matter volume, whereas when partitioning older subjects by memory performance, there are no significant differences for gray matter volume, but strong differences for memory-related brain activity. So that's it. And if this has made you curious, feel free to have a look at the paper.